I guess for the overview, that this will be fairly broad in general. Certainly feel free to ask questions as we go through if there's anything you want to clarify or if you do want some more specifics at the time, and hopefully we'll get some time at the end to discuss some of those aspects. So in the spirit of being a little bit interactive, don't get scared everyone, I'd be interested to know out of the people here today, how many people are sort of actively working on biotherapeutics sort of in the clinical space, so anything related to clinical trials. Go on, yeah, hands up everyone. Go on. I can see quite a few. Oh, fantastic. And then how many in the sort of preclinical space, so with animal models, that kind of thing. And that's great. And how many people who are not working on either of those two things, but are just really interested in immunogenicity? Fantastic. Some of you guys as well. And the other question, which I tend to ask around Pfizer quite a bit, is there's a lot of interest in immunogenicity, and often from people who don't have an immunology background. So can I just have hands up from the immunologists? Great stuff. And hands up from the non-immunologists. I'm not trying to single you out, guys. It's constantly a learning experience, and that is what research and development is. So, it's great to come back to this seat of learning to be considering some of these issues that we have. So, without any more ado, but I might get you to do something else later. So with regard to immunogenicity biotherapeutics, why do we care and where do we stand? Because quite a few years ago we didn't care so much, and we're beginning to care, and that might be for a whole number of reasons. So I'm just going to go over a little bit about sort of biotherapeutic innovation and a little bit of background, and then look at why do we care, particularly to do with the impact of immunogenicity. As Nick suggested, that this isn't just, we've seen it in the clinic, it's a problem now. Actually, from our experience, we see quite a lot preclinically, and we get to the point that, wow, it's, that's not just because we're putting a human antibody into a monkey. There's something else there. So we want to understand that before we go and do our clinical trials, because clearly the cost associated with a clinical trial is phenomenal compared to trying to understand what the problems are before you get there. The where do we stand bit, clearly you've got to have a way where you can try and practically manage immunogenicity. So taking a risk-based approach is certainly a good starting point. And when you consider some of those risks, the kind of areas we're looking at are things to do with the product, with the patient, and then with the way that you're delivering the treatment. And these are all kind of things that lead to that, or can potentially lead to the anti-drug antibody production, which is ultimately what we want to stop. Maybe if there's a few things around the edges that you get some response but you can live with, that's not such a bad thing. But as again Nick said, that we want to get to a point where we've got low acceptable immunogenicity. It'd be great to have no immunogenicity, but perhaps that's dreaming a little bit now. And I've just got a couple of slides at the end on sort of future perspectives, certainly is the kind of thinking that we're going through right now. And I guess that will always evolve, so we'll see how that evolves over time. Just a bit on biotherapeutic innovation then. I guess what we're kind of over 30 years or so of doing this kind of work, not me personally, and we started off with things like recombinant DNA, so expression of essentially endogenous proteins or replacement factors. And as we went through, I realise this is stuck on, hasn't it? It's your dangerous tool, Jeremy. I might try not to shoot myself with it as well. We can see with this one. So going through from, I guess, chimeric mouse antibodies, humanized antibodies, we've started to do some funky things with the Fc fusion proteins, but of course, if this is like a receptor on the end, an endogenous protein, you're probably still in fairly good shape. And so this is where innovation kind of helped from the point of view of immunogenicity. We got to fully humanized antibodies, and that's kind of a nice place to be. Had we left it there, we might have been a little bit happier about the world in terms of not worrying about immunogenicity so much. But then we start adding things onto our antibodies, coming up with novel scaffolds. The biosimilar world, certainly an area now that due to changes in regulation, particularly in the US, that this is really opening up, and understanding what biosimilar really is something that's still It's probably going to be a legal debate, unfortunately, or fortunately if you're a lawyer in the area. But also then moving into this area of bio-beta proteins. So they look pretty much like these, but there's something better about them. And certainly my belief that immunogenicity is going to be a key feature over here. So for the products out there where we have immunogenicity issues, which is quite a lot of them if you consider that these things often offer chronic disease and are dosed over vast time periods, that if we can get to a point where we've got lower immunogenicity products, then we're going to be in a better space. I mean, the other thing as well that we need to consider is that there's different ways now that we've gone from giving things as IV infusions, which certainly from some of my background in vaccines, you can be sure that if you deliver something IV, you're going to get a much lower response than if you start doing something like giving it through subcutaneous administration. And also then some of the alternative delivery routes that people are looking at, whether that be kind of inhaled protein or different forms that might be delivered. So there certainly are things to consider as you kind of go through this process of evolution, and it's kind of bringing the immunogenicity question to the fore. So what's immunogenicity? So I guess historically, we kind of put protein into people and we looked at what came out here, so the secreted anti-drug antibodies, and we kind of, this is either a person or an immune system. And this was kind of acceptable for a while, but given that we know quite a lot about what's going on in this black box, we really do think that we can start to affect some of how we actually get from the process of protein to anti-drug antibodies. And I don't want to go through this in great detail. For the non-immunologists in the room, this is a simplified model of the immune system. It may not look so simple, but essentially to say that while there's been a lot of focus on sort of the B cells because they're the ones that actually produce the anti-drug antibodies, clearly the T cell help is a key feature, and of course we'll hear a lot more about that today. So in terms of where we are now, it's very much kind of trying to understand immunogenicity from the point of describing what we see and then managing that risk. So there's quite a lot of effort gone on in terms of the regulatory world to actually understand, as Nick says, is this a safety issue? Is there a way that you can mitigate that risk of your safety issue, and manage that safety issue to a point where take replacement factors for haemophilia. Your safety issue is as much the fact that if you put the protein in and it's cleared rapidly because you've got anti-drug antibodies, then you're not having an effect on the disease. And so your safety issue is that you've gone back to not covering the disease. But we do want to take things on a little bit from this. So, I think there's a couple of crystal balls in this presentation, but do we want to get to a point where we're able to predict what's going to happen? And at least we want to be in a position where we can say with some certainty what the problems are, and then actually work out how we can mitigate against those problems. So I've got the old black box again. It's the easiest way to show the immune system. So why do we care? Well, we're putting in these sometimes funky molecules into our immune system, and we're getting a response. And again, the issue that with safety, this may be something as simple as a hypersensitivity reaction. It may be something more complex, and certainly in the preclinical space, we quite often see problems with immune complex formation, which would lead to termination of some of the animals in the studies. So that's clearly is an issue that's then flagged as a potential problem for taking things into the clinic if you don't have enough understanding of how that was formed and why you got that result. You may have noticed I'm in a department called Pharmacokinetics, Dynamics and Metabolism, which is partly sort of historical from Pfizer's setup. But the reason that Pfizer decided that immunogenicity, at least for the preclinical and early development space, should sit in this area is that actually the efficacy issue is really the big one. So understanding the pharmacokinetic, pharmacodynamic relationship is something that we need to get a good grasp of. So this is kind of schematic here, just kind of shows with the presence of the anti-drug antibodies that often you end up with clearance of your drug. And again, whether in the clinical space that means you're not treating disease, or in the preclinical space it means that perhaps you can't run a study that for long enough to actually see the effect of your drug. It's still like a serious issue whichever way you look at it. So if we can get over our immunogenicity issues early on, we can start to understand things that are helpful for all stages of development. So this is just really a look again historically, and one of the key features is that we can consider all biotherapeutics to be immunogenic. There may be one or two that haven't had any immune responses observed for them, but on the whole there's always some level of immunogenicity. And it kind of follows through that it's not necessarily always down to the product. So you're putting these products into different patients who have different disease positions. Clearly we've all got different genetics. You know, we're not quite as inbred as some of our mouse strains. So there's things that we need to understand about our patients as well. But just really very quickly to pull up that there are some cases of Xiaflex here, which is a collagenase, and although there's 100% ADA response, that there's no effect on efficacy. So you can go ahead and use that, and you could argue immunogenicity is not a problem in that space. Whereas for things like the factor VIII, if you've got neutralizing antibodies, sort of historically termed inhibitors, then you get clearance of that protein and you just lose the effect. So there's just a few examples there. And so really kind of it's not only because we can measure these things that we want to now measure them, but that we do understand that we have a need for change. So if we could work out which products are likely to cause immunogenicity or anti-drug antibodies, that we can start to sort of focus our efforts around sort of mitigating against that risk and either change something about the product, be it its sequence because it's got a strong T-cell epitope. We may have to accept that subcutaneous delivery is out for some products and that our best way of actually getting that product to a patient is to deliver it intravenously, although you know, it may not be so convenient. But we want to understand this early on so that if we do make any changes along the path of development, and one project I'm working with at the minute, we're coming towards the end of a phase 2 that's looking very successful, and they're making their plans for a phase 3. But there's also a change in manufacturing going on in there. And we don't really want to go into a phase 3 and just do all of this work and then find out, oh, it's that manufacturing change that causes a problem. So we want to understand what the risk looks like before we go ahead and with that new material. So there's all these things that could change along the way. And we've got to understand what our black box immune system is doing so that we can try and avoid this ADA response in the clinic. So clearly, when you're working in business, you have to understand something about the numbers going on. So cost of development of a new biotherapeutic. This is actually I guess 2007, so we can be pretty sure that this is a higher number than this nowadays. And again, if you've got to the point of a late stage clinical trial before you pick up a problem or before you introduce a problem, that can cost you quite dearly. On another level here, a different way in terms of looking at when immunogenicity occurs. So things like the replacement factors, that there are sort of the immune tolerance induction methods. And these can be pretty costly to actually get to a point where you're causing or allowing patients to become tolerized. And therefore, clearly these are the sort of figures you want to be avoiding. Okay, you might have sold more product by doing it, but if you enter into a market where the payer understands that they might have to pay this amount out if there's a problem, then they'd rather go with someone else down the road who's not going to have this problem in the first place. I won't say very much about the regulatory guidance, apart from there's quite a lot of it. It's not all in one place. And I think this afternoon we've got Robin talking about aspects of interactions with regulators. So I will leave that for this afternoon. It's certainly not an area where I'm an expert either, so I'll be happy to talk at any point about some aspects, but there's probably already people in this room who know more than I do on this. Okay, so kind of going back now to 2004, which is before I got into this field, there was already some fairly clear guidance, and there was a couple of papers, or two-part paper, looking at risk-based approaches to immunogenicity. And I guess this comes out of some of those, I guess, immunogenicity issues that were pretty serious. So I mentioned the factor VIII, but things like erythropoietin actually led to severe safety concerns, and there were issues around that. So this was not just that the sequence of the molecule had changed, because it kind of hadn't. It was administration route, it was things like removal of human serum albumin and inclusion of tween 80 that actually seemed to be the factors that led to those immune responses. So at that time, there was already a need to try and understand all of the risk factors in order that you could then try and mitigate against these. So I guess the bottom line, and again, Nick certainly mentioned this, that if we understand this risk, we've got sort of knowledge of what the risks are. So essentially, how's the immune system working? What's it got to deal with? But also then the new tools, which again, I'll touch on briefly in this presentation, but we'll hear a lot more about during the day. So if we've got an understanding of, or if we're building our understanding by applying the new tools that we have, then we're going to be in a lot better position to understand what risk we're carrying. So this is kind of a selection of risks. So we have a process within Pfizer, and kind of having come from very much a discovery background myself, I'm not always a fan of process because it sometimes closes you in a bit. Having said that, clearly when you get into the regulatory space, you need to, everyone needs to be clear what your process is so that you've got everything covered, and that you can start to lock down on some of these things, or at least have an understanding of where you sit. So I've already mentioned that we have risk factors associated with product, with treatment, and with patients. And this is kind of a selection of some of those things. So whether it be down to foreign sequence and the T and B cell epitopes, the product biology, so things like risks associated with binding to antigen-presenting cells, whether it's the pharmacology of the drug. Now, we do a lot of work in inflammation, autoimmunity, and forever I'm speaking to project teams who are saying, well, our drug's designed to lower immune responses, we're not going to get an immune response to it. And it's like, well, really, how confident are you that, well, one, you're going to completely reduce the effects of the immune system, and also then to new, I guess, new entities the immune system's seeing. Because if you're going to completely ablate the immune system, you're just going to get people with lots of infections, and it's not going to be a product. So there's often a little bit of education about exactly what the drug will be doing, which, again, sometimes I'm coming to people very early on in their projects, and they're quite receptive to the idea of this. Some people are very well set on, I know what I'm doing, and immunogenicity is like a bit of a fly on me. I'm trying to swat it away and not worry about it. But certainly we do have some examples where that kind of swatting things away and then actually seeing in a clinical trial that you've got massive immunogenicity issues is starting to change things, certainly within our company. So with regard to treatment, things like route of delivery, dosage, duration. So do we understand that if we're giving single dose of something, which is fairly rare for a lot of our projects, that we're probably in good space. If we're giving chronic administrations, then we really need to understand a little bit more about how immunogenicity will progress. And certainly the clinical experience does seem to be that you're not necessarily going to get a strong immune response after 1, 2, 3 doses. We're not vaccinating. But sometimes the immunogenicity, the anti-drug antibodies, become apparent over a lot longer time frame. And again, this is why you wouldn't necessarily see things in a first in human, and you have to understand what your risks are. So I alluded to this earlier as well, patient population. There may be some things as simple as genetic linkage, which is associated with your HLA type. Now, if you've got strong epitopes for those HLA types which are associated with a disease, then you've probably got a better chance of driving an immune response to it. So these are things that, maybe not on all diseases, but certainly they are things worth looking at. And another area we're looking at as well is this pre-existing reactivity. I was calling this pre-existing antibodies, and our assay group were very cautious about this and saying, well, we don't know for sure that they're antibodies until we've gone and done the confirmatory assays. So this is kind of another point about those assays, that if we don't know exactly what we're measuring, we have to be a little bit careful about it. But we are seeing more and more that there is this pre-existing reactivity, and I've got kind of a slide later on this. But all in all, these are your immunogenicity risk factors, and we've got immunogenicity risk, and we actually call it immunogenicity risk and mitigation plan, or the IRAMP. And the number of times I've seen people do a little I on the front of that, they say, we're not Apple, guys. But it's certainly something that if it sticks in their mind, that's a really good thing, because even if you do nothing else, you've got to do this. You've got to understand, what are your risks. This slide, there's quite a lot of words on this, but it does really just back up what Nick was saying, that you can do things early in discovery. Can you pick a candidate that's got a lower risk of immunogenicity than something else? And again, speaking with project teams, it's often, these are the issues that we have are related to potency or our ability to produce a lot of an antibody in the favoured cell line. And immunogenicity does always seem to be a bit further down the list of important factors, but we're trying to raise that up. And if you can work out what factors are most likely to contribute to risk early on, then you've got a better chance, as I mentioned before. If you have got a strong T-cell epitope in there and you think, well, actually we need to change the sequence on this, you've got to know early. It's no good going in when you've started your clinical phases and say, I think we just need to change a couple of amino acids in this, because that takes you back 18 months whilst you go and prepare all your GMP material for the clinical trials again. So some aspects of non-clinical development. One key area is the understanding about whether your anti-drug antibodies are going to affect the results from that non-clinical study. So if you can't interpret your study, then you're going to have to redo that study. And again, this is something that's happened a couple of times for us. And you would lose several months in the development process there. So that's obviously not ideal. With the early clinical, say the pre-existing cross-reactive antibodies, is something that we want to understand, and as to whether is it just a reactivity that's is cross-reactive, but you're not going to generate any more when you go into patients with your drug. Things like what consequences and what mitigation. And clearly with the later clinical development, understanding any changes you make, if you've got an idea as to what effect those changes are going to have, you're in a better position to actually observe them in the clinic if they do occur, and therefore be in a position perhaps to not necessarily just change something back, but make other alterations that would give you a better final product. Okay, so in terms of immunogenicity risk factors, I've kind of put the slide up, which is by no means comprehensive, but just sort of thinking more broadly about risk factors, that in an ideal scenario, you put your protein in, and there's such a big sort of activation energy, if you like, to get to a point where you're producing antibodies, that you just don't see a response. And this is kind of our ideal scenario. We've either kind of developed a protein with no risk factors, or we're able to mitigate against those risk factors, that it's really difficult to generate a response. So this is ideal, not reality. So the reality is, you might have 1 risk factor, 2 risk factors, 3. And I wouldn't like to say exactly how much each risk factor drops this barrier down. Suffice to say that if you've got enough of them, you have what we call a vaccine. So that's so much easier, that. Some of them vaccine colleagues would disagree, but certainly this is an area where understanding how many of these risk factors, how they interact, is going to be really key. So we go from low immunogenicity to high immunogenicity. Here's, again, another list of those risk factors, which I'll look at some of those in more detail. But I think the key piece for us is knowing how to integrate all these risk factors. I would say that probably as a scientific community, it's a question we've not really quite got to grips with yet. And there are efforts, quite a lot of efforts focus in one or two of these areas, how you bring the areas together. I think the big challenge for us is to get to a point where we can bring all of these things together. And that's not necessarily in one experiment, or is that a clinical trial? Oh no, that's not an experiment, is it? So I think we should understand that our ideal scenario is that we could do enough work before we go to the clinic that we do know how to integrate all these factors and that we can truly predict whether we're going to have an issue. Okay, so some examples of those risk factors. So I've touched on some of these. So in terms of the foreign sequence risk, certainly if you've got a deficiency in a replacement factor, a lot of these are genetic deficiencies which would completely ablate the protein or terminate the protein early, which would mean that you've got effectively a significant amount of foreign sequence in your therapeutic. And I discussed some of these before about whether you've got chimeric mAbs. Arguably even a fully human antibody, it's got CDRs which the patient wasn't producing initially. So I think you do have to consider that as a foreign sequence risk. There is some active discussion in the community about how we generally handle the foreign sequence within our antibodies, whether there's aspects of tolerizing to antibodies generally, or whether there's sort of control through sort of anti-idiotype type antibodies. Certainly an area where there's quite a bit of development occurring. Some of the pre-existing anti-drug antibodies, whether that be to things like the carbohydrates, which are not of human origin, certainly host cell line proteins can be a bit of an issue. I guess some things like autoantibodies. Well, this prior exposure to homologous product is certainly something that I think we're beginning to see a little bit in our phase 1s, and there may be an element of going to normal healthy volunteers who happen to volunteer for all sorts of different trials. And I'm not sure we're that good at really knowing what they've had administered to them. And from speaking to some of our clinicians, the patients aren't really that good at knowing what they've had, so there's no point asking them what they've had. And I think to some degree we probably should be pretty good if we're continually dosing them with things. But if they're moving around and being dosed by different companies and that, I think there is something that we need to understand a bit more about, and maybe we do need to be a bit more cautious about how we're selecting patients, or even the normal healthy volunteers, sorry. So a couple of other things on here, say the route of administration risk with the EPO. Aggregates and formulation often come up, and aggregates, I guess our pharmaceutical sciences group are very keen on understanding aggregates, because if you've got above a certain threshold for any given protein, that you end up losing out on your stability, because those aggregates start as nucleation points. And I guess for a typical product, we'd want to be able to get a 2-year shelf life. If you've got significant amounts of aggregates, just the whole thing will end up deteriorating over time. Having said that, the figures they often use would be sort of in the 1% or lower. And if you consider actually how much protein that is for some of our therapeutics, that you're certainly looking at the potential of dosing sort of high microgram, even sort of low milligram amounts of aggregates to patients. And again, from my understanding of a lot of vaccines, you're in a good area that if that is sufficient to drive an immune response, you could get a pretty good response. So we do need to understand this a little bit more, and some of the context about that as well I'll come to. So to look at some of the risk factors, I've got T-cell epitopes here. And this is actually a paper out of Abbott, so Fiona Harding published this in 2010. And they were looking at here, just to take you quickly through, on the left-hand side, individual data points. So these white circles would be the framework regions of antibodies, which generally aren't very strong stimulators of T cells, but where you've got the CDRs, you get these much stronger responses and more frequent responders, and this is kind of the average data for that. So obviously with these kind of assays, it's deciding where your cutoff points are. Same for the anti-drug antibodies, really. You know, the whole field is, the assays are quite key to being able to understand the data coming out of them. But suffice to say that there is quite a big step up when you go from having sort of self regions, if you like, which I don't want to say for sure that framework is always self, because clearly we've got polymorphisms, and so the framework that we decide is suitable for most of the population may not be for either one particular population or certainly individuals within the population. But certainly the CDRs come out as being of higher risk. So that's where, when you're thinking sort of more broadly about your product, where you would see the greater risk being. So this is just a look at some of the pre-existing antibodies. I've already gone through a bit on in terms of the sources. One of my colleagues, Li Zhu, had got involved with the American Association of Pharmaceutical Sciences group in this area. And they actually looked at, I think it was something like 40 clinical studies with 13 biotherapeutics. And this was, I guess, 6 of those biotherapeutics. There were pre-existing antibodies in the study populations. So again, that may not necessarily be a problem, because the result could be there was a pre-existing response and the response stayed the same, which meant that it was just something cross-reactive that you weren't stimulating any further. But it is a bit of a risk. If you ended up with very high levels of this as being pre-existing, you're going to have to start getting into understanding whether you've fundamentally changed that immune response if you do see an increase in the response. And things like characterizing your antibodies to the point of understanding which epitopes they're binding to is actually quite a big job. Having previously done some epitope mapping, it's kind of, yeah, it's interesting. It's on the whole quite academic, and it's not something that we want to be getting into for every product. We'd much rather be able to avoid this as an issue. So route of administration, touched on this as well. I think that one of the key things here is that our main drive for products is to get more to subcutaneous delivery than for IV delivery. Associated with that then is the general feeling that subcutaneous route is more immunogenic. So that may not hold true for all products, but from a lot of our preclinical studies and I guess most of our clinical studies, it does hold true. So you have to start thinking about, well, what are the reasons behind that? Because whilst on the one hand we could say, oh, well, we'll just deliver everything IV and we won't actually make it easier for our patients. If a competitor company says, well, we know what the problem is and we're going to fiddle around a little bit and make sure we can deliver it subcutaneously, then the chances are their product's going to do better than your own. So we want to understand these risks. So these kind of mix in with things like, well, if you're delivering aggregates into this space, again, I think that's a good vaccine. So it's a way to think about how these things interact. So there's certainly some literature about potential direct activation of B cells because you're potentially presenting an array of epitopes to those B cells. Might depend a bit on how your aggregates form. Certainly the localization effect, again, from the standpoint of being a pharmacokineticist as well as an immunogenicity person, the pharmacokinetics aspect is often quite good from subcutaneous delivery, that you get that depot effect and a slower release. But if you're hanging around in an area where you've got a lot of immune cell surveillance, then potentially that's bad from an immunogenicity standpoint. Some of the other things linked with some of the I guess this is more linked with the aggregation and route of delivery would be things like potentially activation of complement in this space. And then that may well lead to the suggestion of you get sort of platelet binding, which may favour uptake by the dendritic cells over the macrophage. Sorry, I've not completed this slide. I was going to put a reference in there. I will make sure I do that for the final version that you guys get. But this really shows that maybe there are some other patient factors that we need to consider. So complement status, be that down to polymorphisms or actually physically measured values, and also how DCs activate. So just an indication there may be a few other things. And so one of those things, I guess, is whether we've got overall this approach reduces the barrier to sort of the T cell responses from because you're able to load that protein more effectively in whatever way into the antigen presenting cells. Again, from the point of view of trying to understand a little bit about risk, one of my colleagues in our pharmaceutical sciences department has been looking at the relationship between what they term aggregation-prone regions, so that's the APRs, so how they align with T-cell epitopes. And certainly it looks like there is quite a lot of overlap with between these two types of sequence. And it may well just be something along the lines of whatever physicochemical properties would favour aggregation formation are also the ones that favour binding within the MHC. So I guess their perspective is they want to reduce aggregation. And our interest is we'd like to reduce T-cell epitopes. And if we can do those two things with maybe a single amino acid change for some of these epitopes, or I guess agritopes as they are often called for interactions with MHCs, that would be a double win for us. So anything that we can make a single change and get a double benefit has got to be a good thing. Okay, so when we think about where do we stand, we've talked a little bit about this already. We want to generate those tools which are going to improve our risk assessment to the point where we can understand that we're taking the right thing forward. Sort of back to Nick's point as well, you can do this at any stage in development. If we want to throw our full weight of assays and capabilities into very early projects where, let's think our Biotherapeutic team who actually make the antibodies, they've just done a phage display library and they've come out with their 100 best hits. Some of it may not be too practical from the standpoint of actually funding that kind of work, as they don't seem too eager to fund all the work to look at all 100 hits. So we're really kind of looking at how we would be able to triage things on the basis of certainly other aspects, such as maybe some of the in silico work around T-cell epitope prediction. I'm still a bit sceptical on the B-cell epitope prediction by in silico. I'd be interested if people have got views on that. But certainly when you go down here, sort of if you look at in terms of getting from something nice and cheap in a computer to a few binding assays, and or you might recognise this from a ProImmune website, the cellular assays, potentially to sort of humanise mouse models, and then obviously into the clinic, because we certainly don't want to be waiting till we get here to do everything, but we do want to try and implement some of these aspects. So when you come to T-cell epitopes, we've got really well-developed tools in understanding these aspects. It may well be that some of these assays can also help with things like aggregates, but there are fewer specific tools in these areas. I think actually that some of the B-cell epitopes, there's some pretty good ways of trying to determine what they are if you're willing to put in the effort to understand those. And ultimately, we think that a lot of these things could be done in essentially a human immune system in a mouse. So the one that seems most favoured at the minute is called a BLT mouse. So that has liver and thymus, and then also CD34 positive cells grafted in. So it's not an easy model, and it's not a human, but it may well be the best thing that we've got as an alternative to going into people right now. So we're actively investigating these areas. And as I say, the one thing that we really are applying is this pre-existing antibodies in the clinical space. In terms of maybe what we should be doing in vivo, and I'll probably come back to this later, but certainly there's some pretty easy things to do. So if we understood the HLA types of our clinical trial subjects, that'd be a good start for us. And a relatively easy thing to do, and we're just not doing it. I say we're not doing it as broadly as we'd hope to. We're just not doing it right now, and it's real kind of bad position to be in when this is something that we should be doing in order to at least get a handle on what patient factors are having an effect for us. So I think we've got a talk this afternoon. Is you this afternoon? No, you're this morning, aren't you? Yeah, this afternoon, sorry. So we have an expert in the room, which is good. And so I'll just quickly go through here an example of where we're looking at the in silico tools. So there's the Immunogenicity database, which is a wonderful repository of lots of data. And that's kind of good in some respects. It may be bad in others that the data isn't really curated, so you can kind of get lots of interesting information, but where do you go from it? It kind of would still suggest that you had a lot of work to do. And again, if you want to try and apply this process to 100 or more antibodies in an early round screening, that's actually quite tricky. We're also using this Epimatrix system from Epivax, and that kind of their big selling point is that they've curated the data in a manner that would allow you to be more predictive. So we've actually looked at, I'm trying to think how many sequences we've put through. We've probably put a couple of hundred sequences through the system, and you get, again, a lot of interesting data. What the in silico is really predicting is just whether a sequence binds to an HLA type. It's not really a true T-cell epitope. But, it's first step in the process. You've kind of got to go from here to go to here to get to here. If you've not, for example, in a lot of cases, we don't actually have the protein in hand as such. We've got a sequence that's come out of a phase display library that at some point will get grafted into an antibody, or a sort of standard framework. So we're kind of keen to use that. And what we've seen so far, we've got data suggesting that from here to here, you've got about a 75% sort of positive predictive value. And it does tend to over-predict in our hands from what we've done so far. So the one key message from anyone who's taking this kind of approach is that sort of any epitope's predicted, so they do need further confirmation. And this would just be a sort of a first-round screening tool. So we'll hear a lot more about these kind of assays today, I'm sure. So I won't go into great detail, but looking at sort of the T-cell assays, whether it's PBMC, where you can either go in with whole protein or peptides, and actually look at this interaction between the antigen-presenting cell and the T helper cell, leading to proliferation of your CD4 T cells. And one of the key things I think about this assay is I understand that if you put some immunomodulatory molecules, say a monoclonal antibody to TNF-alpha, for example, in this assay, that's really good at mopping up your cytokine and preventing the proliferation. So you might put in something that's really immunogenic, but you wouldn't see anything in that assay, which is why this assay is then used, which is the DC T-cell assay. So you can put your therapeutic protein in with the context of your DCs away from your CD4s, and then you bring the two together, and then you can look at your expansion of T cells from there. So I guess we'll hear a bit more about that later on. So in terms of decision making, we want to be in a position where we can rank some of our leads. It's not always 100. My job isn't always that difficult. But we do want to be able to select early on. There may be opportunity for re-engineering, as I said before. I'd much rather do this preemptively because although remedial in theory is possible, I would say that would be a unique case rather than the norm. But immunomonitoring, this is something that we really should do a lot more. So understanding what the non-clinical study design looks like, clinical trial assessment, I was supposed to say, and selecting the right patients is a possibility. I do get quite a bit of pushback on the selecting the patients, because there's been times when I've wanted to exclude patients based on their HLA type because of a risk we've observed later on. And that seems to be not very well accepted right now. I would say that there would be an argument if you had a molecule and you really believed that you were hitting the right pathway and you were a leader in the field, to go and do your proof of mechanism, potentially even a proof of concept study, and make sure that you're doing everything you can to get a less immunogenic backup in line. That's my opinion. Not many people have taken that on board and actually invested in that right now. So on to the future perspectives. We kind of are constantly reinventing management speak in Pfizer, as anyone who works for a big company will understand. A few giggles there. So our current initiative is we've got a journey to the 3 horizons. So I'd like to take you all on that journey with me. It's pretty simple, and it's not something Pfizer have invented. We've put a spin on it. We're trying to get everyone behind it. But the simple thing is that there's the stuff we can do now, there's the stuff we know we can do in the future, and then there's the stuff we'd love to do in the future. We might not exactly know how to do it right now, but if we can work our way through that process, eventually what we have on our 3rd horizon will be the today. Again, for those who work in a big company, that's ideal if that's like in 3 years' time rather than 20 years' time. What we're looking at is, now, broad application of available tools. And this is, it's really easy to sell the immunogenicity and risk and mitigation plans to people. They need to spend a bit of time looking at their product, looking at the literature, understanding the patient populations, all things they're doing anyway, and just bring that information all together. So we are applying these. We're using these as basis for putting information into INDs, for example. The pre-existing antibodies we're using a bit on a case-by-case basis right now, as I say, in the inflammation area, that we're, I guess more the autoimmunity area, that because of things like rheumatoid factors, it's really good to understand any pre-existing antibodies to your product. And I say the T-cell epitope assessment, we know the tools are there. We're trying to convince people to use them. Getting things like the HLA type data is a real good start. And ideally, where we see responses that we want to understand a bit more, going on and doing the T-cell, looking at what those cognate T-cells are is also would be an important feature for us. So the new generation of tools is our next horizon. And this is to get to a point where we can get a model where we can start to bring some of the risk factors together. As I said earlier, I think a key thing for us is to try and understand how all the risks integrate. And it may be difficult to do that in the context of a test tube. There are some efforts underway on in vitro systems like the artificial human lymph node. But these are really areas where we need tools that are going to allow us to assess multiple risk factors in one go. And way off in the future there, maybe 3 years, maybe a little longer, that we want to try and integrate all of these tools to a point where we could understand our sequence, understand our patients, understand our ability in terms of maybe how many aggregates are present, whether we're having any pharmacological effect on our immune cells, and pull that all together. And again, historically, the perspective of my department is we've done a lot of stuff on small molecules, and we've actually written a book, not me personally, but we've written a book on how to understand pharmacokinetics and dynamics and metabolism of small molecules. And our management would love to be writing the book on the large molecule assessments, which includes immunogenicity. And where we got to with the small molecule was to be able to predict a lot of this based on in silico, so that we can take our massive compound libraries and really select down to those key leads for us. And actually the attrition due to these kind of risks has been massively reduced. So there is willingness and a belief that it's capable for large molecules as well. And just to give you a quick idea of where we are with this, there's something called the FIRM, or the Fully Integrated Immune Response Model, which our teams are working on. Now, there are a number of other groups working on these kind of initiatives, but essentially it's like, can you build a model of an immune system? And with any good model, you're putting in as the sort of minimal number of factors that will give you an accurate result. So that's quite challenging along the way. But we're building on what was already out there. So things like a TB model with dendritic cells, T helper cells, macrophage, tumour model that obviously has a tumour effect and cytotoxic T cells, and then the antibody production via the B cells. And clearly, to incorporate all our risk factors, we need more than this. But this seems to be a reasonable predictor based on data we've seen. The challenge always with a model is to be able to produce a model, make some predictions about what might happen, and then when you see something different in the clinic, you improve your model on it. Where we face the challenge is that people want a prediction to be right. So we are a little bit cautious about saying we have this now. This is why this is very much in the future. We anticipate that we will get to the point where we know what our product will look like. So I think with that, I'll go back to the crystal ball picture. Just like to thank here Bonnie Roop, who's heads up our Immunogenicity Sciences group. And you saw her picture on one of Nick's slides as she spoke at the meeting in Boston last year. Lee Zhu has been doing a lot of work on the pre-existing antibodies within the context of the AAPS group. Xiaoying Chen and Paolo Vicini are working on the modelling side of things. And from our safety group, Deborah Fink actually drove quite a lot of the work behind the in silico assessments. And Sandeep Kumar is our pharmaceutical sciences immunogenicity and aggregation type expert. So I'd like to once again thank ProImmune for inviting me to speak. And I'll thank you all for your attention. I'll take any questions.